Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is December 4th, 2025, and our guest is Mike Beckstead. Mike is professor and Hille Family Foundation Chair in Neurodegenerative Disease Research at the Aging and Metabolism Research Program at Oklahoma Medical Research Foundation in Oklahoma City. Mike's lab focuses on the midbrain dopaminergic neurons in the ventral tegmental area and substantia nigra. These are the neurons that provide dopamine to practically the whole brain, especially the forebrain structures that are really important in motivation. We've talked a little bit about dopamine in previous podcasts, but Mike's expertise is especially strong in the area of the molecular mechanisms that control electrical activity and synaptic transmission by dopamine. So these are the cellular processes of dopamine rather than that network or genetic things that we often talk about. So hi, Mike. Welcome.
Hi, thank you. It's great to be with you here today.
So Mike's also a former colleague of ours, and I'm surprised. I was surprised to discover today that hasn't done a podcast with us before. He's been a participant in podcasts, but never been the center of attention.
That's right. If you listen through all the old ones, you'll hear my voice every now and then, but I've never been the guest on it before.
I couldn't really believe it. I did an exhaustive search this morning because I'm sure that we must have done a podcast with you. So it's a failure that we did, and I'm glad to.
We're going on, 18 years or something doing these?
Yeah, long time. Sorry. So also with us is Matt Wanat, our local dopamine expert and many-time podcast participant.
Howdy.
Hi, Matt. And I'm your host, Charlie Wilson. So, Mike, when we think about Alzheimer's disease, we normally think about the loss of neurons in the cortex, in the hippocampus. That's what we're usually talking about. And there are cognitive and memory deficits that seem appropriate to be caused by those cell losses in those places. But there are also other symptoms that don't seem to be obviously the result of cell loss in cortex or hippocampus. Motivational issues, sometimes psychiatric issues like apathy and moodiness and irritability. And those sound like dopamine things, at least to me. Mm-hmm. So is there a dopamine neuropathology in Alzheimer's disease?
Yeah, I think there is. I think for a long time, people may have dismissed some of these symptoms like moodiness and irritability as being secondary to somebody who's losing the ability to function and maybe doesn't have the memory they used to have. And of course, that's going to be frustrating. But these symptoms, which are referred to as neuropsychiatric symptoms, is what they call them. They really aren't a bug. I think they really are a feature of the disease. These things like you mentioned, moodiness, apathy, also anxiety and depression, they're not just in a few or some of the patients, they're in 97 or 98% of the patients. And so there really does seem to be some sort of underlying cause for them other than just a side effect of other things like loss of learning and memory. Something I didn't know until I started studying this is that Alzheimer's disease is actually often comorbid with Parkinson's disease. So Parkinson's disease, as you know, is sort of another neurodegenerative disease that's more traditionally associated with dopamine, right? And what happens is that with both Alzheimer's and Parkinson's diseases, especially as they progress and get to late stages, you can kind of start to get a blurring of the lines, especially if you're looking at clusters of symptoms. And so with Alzheimer's disease, a high percentage of patients eventually end up with some sort of movement problems or some Parkinsonism eventually. Likewise, the corollary of that is that Parkinson's patients, something like 80 or 90% of Parkinson's patients end up with sort of a late stage cognitive disability. And of those, something like 30 or 40% meet the criteria for Alzheimer's disease. And so you've got this sort of blurring of the lines and the mixing. And it might not be really useful to think of, okay, well, Parkinson's, dopamine, hippocampus, Alzheimer's, definitely with brain things, it's not that simple. There's other things we've learned more recently. We know that there's a decrease in midbrain size in Alzheimer's patients, and interestingly, the decrease in the size of the midbrain is predictive of patients who have mild cognitive impairment. So sort of the transition state before you get to full-blown Alzheimer's disease. The patients with mild cognitive impairment are more likely to move on to full-blown Alzheimer's if they have more loss of size of their midbrain.
So when you say loss of size of the midbrain, you really mean this in a sort of gross anatomy sense.
Mm-hmm.
The nucleus itself is shrunk. It doesn't tell us that there are fewer neurons or fewer synapses, or we don't know why it shrank. It just shrank.
Exactly. And, we can do brain scans in living humans, but we only have a certain amount of resolution to that. And so you can detect changes in growth size of areas. You can detect changes in maybe glucose utilization, sort of at a low-ish resolution, certainly not down to this, like the single cell level, like we can get to with some of our animal models that we'll get to.
But if you look post-mortem at the substantia nigra of a Parkinson's patient, you can really see that there was a loss of neurons. Is there any comparable neuropathology that goes with Alzheimer's disease that you could see in a stained tissue section or something like that?
I think there is. I don't think it certainly hasn't been looked at to the level that it has in Parkinson's disease, but I think it's widely accepted at this point that you do get some dopamine neuron death in Alzheimer's disease. Now, realize also that doesn't even really make it special. You kind of get shrinkage of the whole brain, especially the really advanced cases of Alzheimer's. The ventricles enlarge in size, and the whole brain shrinks down. And so it's not really a surprise that you get neuron loss in sections. And it doesn't necessarily mean that it's special.
Yeah, people say we're losing our neurons all the time, and
right.
So just yesterday, I had more dopamine neurons than I did today. How about something else? Like, can we just measure the amount of dopamine in Alzheimer's, or some kind of measure of dopamine function in the nucleus accumbens or striatum or something?
Yeah, you can measure. You can definitely measure connectivity using MRI, and you can measure the sort of the functional connectivity between brain regions, and there does appear to be a loss of connectivity between specifically the ventral tegmental area dopamine neurons and the targets for that, which include the hippocampus and cortex, by the way, and a little bit in the substantia nigra, ones that project to the dorsal striatum. That's more of the motor pathway associated with Parkinson's disease. Both functional connectivity. You can also measure a dopamine transporter signal through PET imaging. And that's reduced in patients with Alzheimer's disease. Again, preferentially in the ventral tegmental area ones that project to ventral striatum, hippocampus, cortex, and not so much the substantia nigra ones that go to the dorsal striatum.
It's hard to just mark that off as generalized shrinking of the brain or anything like that.
Right, there's specific deficits.
When do these changes sort of happen within the sort of typical progression of Alzheimer's disease? Like, when do we start seeing in patients? Is it early stage? Is it maybe an early diagnostic marker of that might precede it? Is it late stage, or does it depend on the individual?
I think right now all we know is that it's there in the late stages. I don't think we really have the answer to that yet. Although, as I think if really dopaminergic things are going on early on in the disease, which I think they might be, we should start looking earlier. There needs to be a way to find, and people are looking for biomarkers all the time, because we're in desperate need of better biomarkers for these neurodegenerative diseases. I think that the dopamine changes, I think you might see them earlier than you'd think, and I don't know it's really been investigated in any detail.
Well, I mean, there's obviously a challenge with human research, because it's hard to sort of interrogate the system there, and you're just sort of getting snapshots based on the imaging tools. But I guess, how can Well, how are you examining this in using sort of rodent models? How can we actually?
Surely there's an animal model.
Surely there's an animal model of Alzheimer's disease. Really, there might be 120 of them. So we were really kind of inspired by a study that came out in 2017 out of the D'Amelio lab in Italy, where they were looking at an Alzheimer's mouse model, an amyloid-only model called the TG2576 model, and they were really this was really the first paper to really identify that there were probably dopaminergic things going on. And so they actually saw dopamine neuron death, specifically in the ventral tegmental area. They saw decreased dopamine release in the terminal field. And they actually saw that some of the behavioral deficits, cognitive deficits in this animal were improved with L-Dopa, which is a dopamine precursor. And so that gave some pretty good evidence that there might be something going on with dopamine, at least in this one model. And so now they didn't do anything with single neurons, and that's sort of what our specialty is, studying single neurons. We do a lot of patch clamping, we measure electrical activity and things like that. And so we wanted to sort of pick up that mantle of this. And so at the time we started the first thing we started doing is studying a mouse model called the triple transgenic Alzheimer's model. And it's an older model. I think 2003 was the initial description of it. One of the nice things about it, though, is it's one of the few Alzheimer's models out there that exhibits both amyloid and tau pathology. And so for an initial set of experiments, while we were trying to see if single dopamine neurons were affected in a mouse model of Alzheimer's disease, we wanted to start with something pretty severe. And so that was sort of our first step. And so
with amyloid, we usually are thinking about a mutation that causes the amyloid to be misfolded or susceptible to misfolding. With tau, though, we normally don't think of tau mutations giving rise to disease. Most diseases don't have a tau mutation, but tau gets hyperphosphorylated or something. So how does the model work in terms of those molecular things?
Well, so the model actually it's triple transgenic because it has three mutations, two of them in amyloid-related things and one in tau-related thing, These are all mutations that have been discovered as part of familial neurodegenerative diseases. And so in the case of tau, which you mentioned, it actually is the P301L mutation of human tau. And so that's a hyperphosphorylatable tau, which would lead to hyperphosphorylation and make it more likely to accumulate in such ways to make these neurofibrillary tangles that form intracellularly and are one of the markers of sort of later stage Alzheimer's disease.
So these are human genes, and they just get gifted to the mouse.
Right.
That's an evil gift.
And then there's mutations in the mutation in the amyloid precursor protein that causes more production of it, and also in presenilin 1, which causes more secretion of amyloid beta, and also sort of promotes the oligomerization of it. And so we have these three things sort of working at once in this mouse model. And it's driven by transgenes by the Thy1 promoter, which is a neuron-specific promoter. And so that's molecularly how it happens with this model.
So that means you get a mouse that looks like it has Alzheimer's disease. I mean, neuropathologically, it has plaques, tangles in all the right places, and
it does. And it's there's a progression to it. And so cognitive impairments, sort of the traditional Alzheimer's-y cognitive impairments with things like water mazes and fear conditioning and things like that appear at around 3 months of age for the mice, which is still pretty young for a mouse. You start to get actual accumulation of amyloid beta plaques at about 6 months, and they get more severe by 12 months, and right around 12 months, that's when you start to get the tau neurofibrillary tangles. And so, there's definitely a progression to it, and some of the behavioral deficits can pop up before you get the gross protein aggregates that you get with the disease.
So, a lot of times we just focus on cells being missing. You lose your cells, and that's why you're the way you are. But usually, probably it turns out it's cells that are still there that are doing the wrong thing, that are causing the symptoms. So how can that be assessed?
Well, the way we approach it is by doing patch clamp electrophysiology in brain sections from these mice. And so you have your experimental mice, which are the Alzheimer's mice, and they're on a certain background, a genetic background. And then you have a control line, which is the same genetic background, but doesn't have the mutated human genes. And so we have two groups that we can compare, and that's how we've done most of our studies to this point. You can take the mice, you can take them at different age points and sacrifice the mouse, section their brain, do it quickly and keep the cells alive, right? And then you can have living neurons that still have all of the experience that mouse had in his whole life is still there, and all the synaptic connections are still there. And you can interrogate that through a technique like whole cell patch clamp. And so you put the section up on a electrophysiological rig. You go down with a really sharp glass electrode. You touch the cell and you measure the electrical activity of that cell. It's an old technique, but it's a really powerful one, and it's one that we still swear by, right?
So the dopamine deficit in that mouse includes some cell loss, or doesn't include any cell loss? Or is it just cells malfunctioning, or are there also cells being deleted?
So what we saw was that we saw a pretty dramatic increase in the activity of these neurons. And maybe that might sound like a surprise, because you think Alzheimer's, you think cells dying, things going wrong, you might see cell firing going away. But in reality, this has actually been seen in other brain regions like the hippocampus as well, is that you can have hyper excitability of neurons that are affected by these disease processes, in this case, these amyloid and these tau genes. So the thing about dopamine neurons are really nice to study. One of the reasons I really like studying them is because they fire autonomously. And so you can put them up in one of these brain sections and they will fire in this rhythmic pacemaker manner that has been studied by many labs over many years. And you can use this as sort of a starting point and then measure how that's affected. And in the Alzheimer's mice, the cells not only fired a lot quicker, but the rhythmicity of the firing was disrupted. And that actually gave us our first sort of hints of what might be going on.
So what causes the cells to fire the rate and rhythmicity that they normally do? Because that's the clue, right? You see something like that happening, and the power of the technique is a lot about why cells fire the way they do. And so you could leverage that to figure out what's going wrong. Right.
In the case of dopamine neurons, and I it's a little bit different for any neuron probably that's spontaneously firing, but in the case of dopamine neurons, there's certain ion channels in the membrane that cause ion conductances, that they're voltage gated ion channels that create sort of this cycle that causes the neuron to go through the cycle over and over again and fire rhythmically. And in the case of dopamine neurons, there are a couple of really important potassium channels that we were interested in. And sort of our first thoughts is if we have these cells firing quickly and less rhythmically, there might be a disruption in one of these two potassium channels, and either the SK channel or the A-type potassium channel. In the case of SK, I'm referring to the small conductance calcium activated potassium channel.
That follows each action potential.
That follows each action potential. So you get a rush of calcium into the cell with each action potential, and that can activate these potassium channels. Potassium channels open, potassium flows out of the cell. You get this really beautiful after hyperpolarization potential in dopamine neurons. And not only does it sort of bring the cell out of the action potential with this hyperpolarization, but it sort of sets the clock, and it sort of resets everything, and then the trajectory starts to head back toward the threshold for firing the next action potential. And so the SK channels are probably the most important for the rhythmicity. Later on in the sort of interval between action potentials, the A-type potassium channel plays a role that we probably shouldn't talk about, because we found that those weren't actually disrupted at all in the Alzheimer's mice. But the SK channels, we found that the currents that are carried by the SK channels were reduced really dramatically in the Alzheimer's line. That was leading to this increased firing.
Made sense that it would speed up if the big hyperpolarization after the spike goes away.
Absolutely.
But why did it become irregular?
I think it's what I said. I think the kinetics of the channel are such that, well, and actually the opening of the channel tracks really well with the calcium concentration in the cell in most cases. Under normal situations, that can be a pretty rigidly set amount of time, and I think that really was one of the things that sets the clock for this pacemaking activity. But what we found actually is that what we believe is going on is that the calcium sensor is a molecule called calmodulin, and what we think eventually determined was that we think the calmodulin's calcium sensitivity is reduced, and that's actually what is making the SK current smaller. And if the cell is no longer able to respond as well or as reliably, I guess, to the intracellular calcium concentrations, that would not only make the fire faster, but it would also disrupt the rhythmicity.
So is that specific then for the SK channel, or is there maybe a more global change in sort of calcium dynamics in the dopamine cells, or do we not know that at all at this point?
I think that's a really interesting question. Certainly, there's lots of things going on with calcium. Calcium does a million things, right? And calmodulin does a million things. It's certainly not special, just affecting the SK. It's possible the SK channel effects we've seen are secondary to something else. But as of right now, I'm not aware of any work that's actually, for instance, measured the calcium signal in the dopamine neurons in Alzheimer's lines. That's one of the things that's definitely on our list. One of the things I want to try to set up soon, do some of that, not only in brain sections, but also in vivo. It'd be really exciting to see what's going on with calcium.
But it would be, it would be interesting if it is very selective for the calmodulin regulation of SK channels, because then that's potentially something that's targetable in a way that maybe you could modulate or reverse these sort of adaptations that you're sort of seeing in the dopamine neuron properties in this transgenic mouse line. I guess along those lines, do you have sort of any molecular clues of sort of what changes? So you've got this transgenic mouse line, you're phenotypically, you're looking at this electrophysiological difference there. And I guess, do you have any insights that, maybe from a transcriptomic level, that these animals or these dopamine neurons from these animals might be different from the control animals?
Yeah. So we did an experiment sort of looking for sort of a molecular explanation for some of the things that we saw. And so we used a technique called patch sequencing for this, which is it was a whole lot of work, but I think it paid out in the end. And as far as giving us directions of things to look at. And so what this is you're doing a patch clamp recording of a dopamine neuron, although you could do it on any cell type, I suppose. You can measure a current. In our case, we measured this SK channel current, which we knew is dramatically reduced in the Alzheimer's line. And then what you can do is you can apply a bit of suction, and if you can kind of get the timing right, and actually what happens a lot of times is the nucleus of the cell will kind of clog the patch pipette, and then what you can do is it turns into a nucleated patch, and you can pull that out of the slice, then pull it out quickly, eject the contents of your cell into a tube that you've pre-made, and then put it on ice. And after you get enough cells, do sequencing, single cell transcriptome sequencing. And what's really nice about the technique, after we troubleshot and actually got it to work, what's really nice about the technique is that not only did we get transcriptomic data, but we got it on cells that we had done physiology on. And so we had these two dependent variables, basically, that we could then correlate with each other. A lot of genes were upregulated in the Alzheimer's line, many more upregulated than down. This is something we've reproduced 3 or 4 times now. But what was interesting is that one of the upregulated genes that was also anti-correlated with the SK channel current was that we saw an upregulation of a kinase called casein kinase 2, which admittedly I knew very little about until we saw it pop up on our gene list. But it turns out that casein kinase 2 is responsible for, it can phosphorylate calmodulin. And when calmodulin, which remember is is calcium sensor for the SK channel, when that calmodulin gets phosphorylated, it actually decreases its calcium sensitivity. And so now we've got a sort of chain of events that we can kind of bank on. We've got an increase in casein kinase 2, and it's causing the phosphorylation of calmodulin, which decreases the calcium sensitivity of calmodulin, which is going to make the SK channels carry less current. And so that's sort of the model that we ended up with through a series of experiments that we did.
Okay. So why isn't that happening in every cell that has SK currents? I mean, I know not every cell has strong SK currents, but many cells do. So what gives it the cellular specificity?
Well,
have an idea?
I don't know that it's not having these sort of effects in at least a lot of the cells that have SK. We do know that the SK currents in dopamine neurons, I think, are pretty big compared to most cell types. And I think in terms of pacemaker firing, I think we know dopamine neurons are pretty, more reliant on calcium-related things than other neuron types. And these SK currents, they really are pretty large. So one of the things we did is we actually looked in a couple of other catecholinergic cell types in these animals. There was the substantia nigra dopamine neurons, which kind of were an obvious choice because they are even more reliant on calcium for their action potentials, and these are the ones that die out in Parkinson's disease. But we didn't see this effect at all. We saw no changes in firing whatsoever.
They do have SK current.
But they do have SK, right? So that's I don't have a great explanation for that, but there was no difference. And then we also did the same experiment in the noradrenergic neurons of the locus coeruleus, also have an SK current, not as big, also pacemake, maybe not as regularly. But there was no difference in firing those cell types at all. And so do I have an explanation for it? Not a good one. But there are definite catecholinergic cell types that have SK currents, and they're not all affected. It just seems to be the ones in the ventral tegmental area.
So a little bit more about the casein kinase too. I mean, this is something I did not know anything about until you started talking about it. How globally is it expressed? And is that potentially a marker instead of like SK, since clearly that doesn't seem to be the silver bullet, but is casein kinase 2? Is it upregulated in hippocampal neurons or other places where you sort of see, is that correlated with a degeneration?
Yeah, so I guess if you're thinking about potential targets for treatments, it's unclear that will ever be useful because casein kinase 2, I think, it's ubiquitous. It's in every cell. And
super important in bone and muscle.
Okay, yeah.
It's so inhibitors of casein kinase 2 are in clinical trials for cancer-related things. I think glioblastomas, and I don't know exactly what it's for, but there's actually 3 of them underway right now. The good thing about that, what it meant is that there are inhibitors out there, and we were actually able to order one of them. And so when we inhibited casein kinase 2, we were actually able to put the physiology of the neurons back, which was pretty cool. It was unclear whether we were going to be able to do that, but turns out the SK channels, they're still there. They're still on the membrane. We actually measured it through immunostaining. The SK channels are still on the membrane, and by inhibiting this overactive casein kinase 2 with one of these inhibitors, we were able to restore the physiology. We did that in brain sections after we prepared them. We also did that in vivo, which was kind of cool. I didn't talk about this in my talk earlier today, but we treated the animals for 5 days, and I think we knew already that compound was going to be able to cross the blood-brain barrier. But when we treated them and then prepared brain sections after that, we had restored the physiology as well. And so, good side, we were able to affect this kinase and sort of put the physiology back, and that's really promising for if really Alzheimer's, if there's a role for electrical and homeostatic disruption, maybe there's things we can put back. Bad news is probably not going to be through casein kinase 2. It affects too many other things, involved in too many things. We'd have to find a more selective way.
So even though we don't know why, it does seem to be the effect is selective for the VTA dopamine neurons. It's not happening everywhere in the brain for whatever reason. I don't know. Maybe it ought to be, according to the way I'm thinking about it, but it's not.
Right.
And so that means that this looking at dopamine cells in VTA is the right place to look. So I'm wondering if you could tell me from what we know about dopamine, because when I first heard this, I thought, if my dopamine neurons fire faster, I get more dopamine. It's kind of like taking amphetamine or something. Shouldn't I be wired? Shouldn't I be more attentive? Why should I be apathetic? Or, right?
Right.
It seems like the symptom doesn't match the syndrome.
Right. And we've done behavior where we've taken these animals and put them through a paradigm where we train them to respond with a nose poke behavior for access to a highly palatable chocolate food pellet, which should be dependent on VTA dopamine. And half of the Alzheimer's mice just didn't get it at all. They were completely unable to learn was usually a very, very simple behavior for mice, in that you need to poke your nose 5 times in a hole for a chocolate food pellet. And so, sorry, I
So
said
is
this
it
background.
possible that neurons are firing? It sounds like the mouse symptoms match
Right.
the human symptoms. Is it possible that the neurons are firing more but releasing less dopamine for some reason?
Yeah, and so that was backwards, right? And it's definitely counterintuitive. And we were talking before about usually increase in dopamine, increase in motivation, increase in behavior. They kind of go together, right? And so this is something we've thought a lot about in my lab, and we've worked on, and we're still getting data on it. But it seems like if I had to put my finger on exactly what's happening right now and take a guess, it would be that I think these cells are so hyperexcitable that they enter a state that we know as a depolarization block, meaning that you get inactivation of sodium channels and they're actually less apt to fire action potentials. We know that dopamine neurons in general can go into depolarization block. It's been speculated that in vivo, some of them in normal conditions do even persist in depolarization blocks. At least that's what some people
colleague
believe.
Dan Latch,
Well, yeah, right.
who was just on the podcast a few weeks ago,
That's right.
has said that in a lot of situations.
Yeah, and I don't know whether that's exactly true under normal physiological conditions, but it does seem like it might be the case. We've shown that these cells are, even though they fire faster and they're more likely to burst, they are also more apt to go into depolarization block when we push them. When we inject somatic current, we definitely see them go into depolarization block in ways that the wild type cells don't. And so if something resembling this is happening in vivo, that could explain what we're seeing, which is this deficit in behavior despite the hyperactivity, too much dopamine neuron activity. Needs to be investigated further.
Somebody should do some fast scan cyclic voltammetry in these mice.
Well, we were talking about doing photometry. So is that the whole story, these intrinsic firing things? Or are there other components that might be also sort of jacking up these dopamine neurons that like, are there maybe morphological or structural changes that are happening here that are making them more amenable to, is it all SK, or there may be other things going on as well?
Right. So one of the interesting things we saw in our initial study, we as part of one of our experiments, we stained for tyrosine hydroxylase, which is, it's the rate limiting enzyme for the synthesis of dopamine. It's a very commonly used marker for dopamine cells, and so we had these stains of tyrosine hydroxylase, and they were decreased in the Alzheimer's animals. But when we actually went in and counted cells, it didn't look like there were fewer cells. So this is using immunohistochemistry, immunostaining. And so, you get these brain sections and you can see this fluorescent signal, and it's lower, but the number of cells isn't down. And so what we were wondering is, we didn't have really super high resolution pictures of this, but we're like, is it possible that we're not actually seeing any sort of cell death, but this decrease in tyrosine hydroxylase is actually due to a difference in morphology of the cell? And so, dopamine neurons have these, sort of broad somatodendritic trees, right? And so that's something we can actually measure. And so we went back and we, using biocytin staining, we put biocytin in our patch clamp pipette. And you break into the cell, the the marker fills the cell, and you can sort of get single traces, morphology of single neurons after that, and image them that way. And so we filled a bunch of cells from control animals, filled a bunch of cells from the Alzheimer's line, and we saw that there was a difference. We saw that there was fewer branches, less volume, less surface area in the Alzheimer's line. And that got us asking exactly what you just said, which is, how is that affecting synaptic transmission? Because for the most part, these excitatory and inhibitory inputs that come into neurons are not coming into the cell body. A lot more of them are coming out onto the dendrites. And if there's fewer dendrites, or they're shorter, or there's less branching, it's possible that synaptic transmission is affected. And so we looked at both. We looked at both inhibitory GABA receptor mediated synaptic inhibition, and we also looked at excitatory glutamate concurrence through the AMPA receptor. And what we found was that we saw sort of a modest decrease in the inhibitory input. So this would be, the input that would tend to stop the dopamine neuron from firing was a little bit lower. And we kind of left with the conclusion that maybe it was this retraction in morphology that was leading to reduced inhibition. The surprising part about this was that when we looked at excitation, we saw not a decrease like you'd expect with the change in morphology, but rather an increase. And not just a little increase. We saw an increase in the size of the currents. We saw an increase in the frequency of the spontaneous currents that are constantly bombarding these cells. We were able to attribute it potentially to both what we call presynaptic and postsynaptic things. So presynaptic things, increased release of glutamate from other brain regions, and then postsynaptically on the dopamine neurons themselves, the receptors, potentially more receptors, or the responsiveness of those receptors. And so there was kind of a really large increase in the excitatory input. And so taken together, you got the intrinsic things we talked about, where the cells are just prone, if completely in isolation, they're going to fire at more action potentials and fire faster, right? But now on top of that, we've got this increase of sort of the ratio of excitatory to inhibitory input. The inhibitory input's down a little, the excitatory is way up. And so the two of those things together are going to work in concert and really could take this from just, oh, look, the cell's firing faster, to this sort of ultra excited stage that could potentially lead to things like depolarization blocks.
So in that case, could you try to rescue the mouse by slowing the dopamine cells down? Like even putting a dopamine agonist on the cell could slow them down a little bit, right?
Sure. Yeah. If
is that how L-DOPA is potentially working, maybe? Is it by having the extra dopamine on there, you're activating the D2 autoinhibitory receptors, and you're sort of like telling the dopamine neurons chill out a little bit. So that then could explain why in the human. So give L-DOPA.
Yeah, and there are a population of Alzheimer's patients that their cognition improves if you give them L-DOPA. Again, suggesting this whole dopamine link. And so if you have increased dopamine release at the terminal in combination with a decrease in the action potential firing, because there's dopamine autoreceptors on the cell bodies and the dendrites, and they're coupled to an inhibitory conductance that we've studied a lot.
Yeah, we know about that because of your work.
Yeah, that was really amazing today. This is the first talk I've ever given where I wasn't talking about D2 inhibitory currents and dopamine.
Well, now we are.
Yes. We're biting the broad.
I feel good about this. But sure, I mean, I can tell you from personal experience, having put L-DOPA on a dopamine neuron in a brain slice is if it's firing, it's going to decrease its firing. You're going to get a sort of a rising tide of extracellular dopamine in the slice, and presumably that happens in vivo as well, and it's gonna put a little bit of a brake on the firing. And if it's already firing too fast, maybe it would normalize it instead of stopping it.
Well, thanks a lot, Mike. That's really cool. I think there's so much to find out about this mouse model of Alzheimer's disease other than tau and amyloid, which is what we normally talk about with us.
Yeah, and I think there's this. There could be situations where maybe Alzheimer's isn't one thing, right? Maybe Alzheimer's is 5 things.
Yeah.
Maybe maybe there's a subset of patients basically that are it's dopamine Alzheimer's, right? And dopamine is actually one of the first dominoes to fall and sets off a chain of events, and those are the ones that might be helped. Whereas there might be another set of patients who have to have a different root cause that maybe it's not dopaminergic so much at all. And so this is sort of what I think about when I'm studying this is not so much that dopamine causes everything. Dopamine causes Alzheimer's, but
it does. No.
I think maybe this is where we end up in 100 years. Is that we're going to potentially find out. Okay, there's the certain personalized medicine things, like there's a subpopulation of patients that we can treat with this, and it's going to help people that have these risk factors, this genetics sort of thing. And as we learn more about these diseases, this is also why it's so important for people to study different parts of the brain and different things that are affected in these disease processes using different techniques. Because I think it's going to take us all pitching in together to really find the ways that are actually going to find really effective treatments for people in a more personalized way.
Wise words. So thank you very much, Mike.
It's been great. I've really enjoyed talking to you both.
And Matt?
It's been fun.
This has been Neuroscientists Talk Shop.